Hey everybody, welcome to video 32. Today we're going to talk about the action potential. Now this video might be one of the most important videos of the entire nervous system because action potentials explain how the nervous system is able to do everything that it is able to do. It explains how neurons are able to communicate with one another and how they're able to carry electrical signals and how they are able to turn stimuli into electrical signals that can be sent to your brain, where your brain then is going to process them and send some sort of motor response back. So make sure that you understand this video and that you are asking any and all questions that you have to be able to describe what an action potential is. So an action potential is basically a term that's used for talking about an impulse moving down a neuron. Some sort of information, electrical signal is traveling down a neuron in one direction or the other. It's going to rely on a lot of charges, positive and negatively charged ions, as well as chemicals, to be able to move information from one end of a neuron to the other. So one thing that is important to know, and as we get out, you'll see here as I go through stepwise on the next slide, is that a neuron has a resting potential or a resting charge. Usually it's around negative 70 millivolts when you compare the inside to the outside, because there are more sodium ions on the outside of a neuron than there are potassium ions on the inside. So if there's more potassium ions inside the neuron than there are outside, there's more sodium ions outside than there are inside. But a lot of components like proteins and things like that inside of cells are negatively charged, which means that the inside of a neuron has around negative 70 millivolt charge compared to the outside of the cell. And note that both sodium ions and potassium ions are positively charged. So this is what we're dealing with here. You can see the inside of the cell is more negative. If we treat outside the cell as 0, that means that the inside of the cell is about negative 70 millivolts right here next to the cell membrane. We're going to see there's all sorts of channels and gates that are going to be responsible for letting things pass through, but we're going to go things step by step in the next slide. So the resting neuron is said to be polarized, meaning that it has a charge. So we saw that resting potential is around negative 70 millivolts. That neuron is polarized until you have some sort of stimulus. Now these stimuli can come in all sorts of different shapes and sizes and different things like that, depending on the type of neuron that we're dealing with. You might have some free dendritic end neurons in your skin that are susceptible to, if they're a thermoreceptor, might be receptive to changes in temperature. They might be an encapsulated ending, so they're going to be looking for things like touch and pressure on your skin. Whatever it might be, there is some sort of stimulus. That stimulus is going to cause sodium channels on the neuron to open in the area that is receiving that stimulus. So if we're thinking like the thermoreceptors in your skin, only the area where there is a change in temperature, hot or cold, is going to be stimulated, and only those dendrites right there are going to have those sodium channels open. What that does is it allows sodium to flow into the neuron. Remember, there's more sodium outside the cell than there is inside the cell, so sodium is going to flow down its concentration gradient. It wants to move where there is high concentration to a low concentration. In this case, they're going to move from outside the neuron into the neuron, and because they're positively charged, that's going to alter the charge that we have on this neuron. In fact, it goes from negative 70 millivolts to a positive 30 millivolts. So we call this a depolarization. We are losing the charge that it has at rest. This depolarization is going to occur only on a certain part of the dendrite that is receiving the stimulus. However, once that depolarization happens, you reach a threshold, generally around negative 55 millivolts. The nearby sodium channels are going to flip open. So even though they didn't receive the stimulus from the external environment, they receive a stimulus from nearby sodium channels that they are changing their charge. And so they're called voltage gated channels. They're susceptible to changes in voltage. They're going to open and allow sodium channels to open downstream, so further down that neuron. And so based on this, we can see how local areas of a neuron can be polarized and then depolarized and cause that information to travel down the length of the axon, because you're going to have an area here become depolarized, and that's going to cause voltage gated channels to open further down the axon, those which will cause those voltage gated channels to open further down, on and on and on, all the way along the length of the neuron. Once those channels are open, what we're going to also see is potassium channels are going to open. However, they are much slower than the sodium channels. So that's why we leave this till step 5 here, is because it happens so slow that we're seeing this depolarization move down the length of the axon before we really start to see these potassium channels open. And because there's more potassium ions on the inside of the neuron than outside, they're going to follow their concentration gradient. They're going to move from inside the neuron to outside of the neuron. And since they are positively charged, that's going to alter some of the charges that we're seeing between the inside and the outside of the cell. And we're going to call this repolarization, because it's going to be trying to return us to this polarized resting state. Eventually, that signal is passed, and you're left with sodium and potassium on the wrong side of where they need to be to get the cell back to rest. And so you have what's known as sodium-potassium pumps that are going to be responsible. They're going to take two potassium, pull them from the outside and bring them back in. And then now that they're pointing inside the cell, they're going to take 3 sodium, and they're going to pull them outside of the cell. And so it's just going to go back and forth until they're able to return the cell back to the charge that it needs to be, right around that -70 millivolts, with the correct ion concentrations of sodium and potassium inside and outside the cell, respectively. Here we can see how things work from a charge standpoint, where we're at resting potential, and then right here we receive some sort of stimulus. Sodium ions start to flow in until we reach this threshold at -55 millivolts. That's going to cause local depolarization to travel down the length of the neuron. We're going to get a huge influx of sodium ions in depolarization. Then we start to have potassium channels open, and they're going to flow out, and that's going to cause repolarization. They do actually overshoot the -70 millivolts, just because they aren't necessarily trying to reach -70 millivolts. They're just trying to move to where there's less of them on the outside of the cell. And so you get this hyperpolarization period, or the refractory period here, where it is very difficult, and in some cases impossible, for that neuron to send another signal, because it needs to get those ion concentration gradients back to their resting point and get back to that resting potential before that neuron is able to send another signal. See if this goes here. There we go. So that action potential that we described there is kind of the general description of an action potential, and that's the one that you most need to know. However, that usually that only applies in unmyelinated neurons, and it's called continuous conduction of that action potential. It is significantly slower than what actually happens in most of the neurons in your body, which is called saltatory conduction, where they have myelin wrapped around them. And so in saltatory conduction, what happens is that action potential still travels, but we get these jumps in between. So we can see this myelin sheath wrapped around, these kind of little cylinders wrapped around it. Remember, myelin is made of fat, which is a really, really good insulator. And in between each of these myelin sheaths, we have what's known as a node of Ranvier. And what happens is, you remember, you get local depolarization in one part, or in this location of an axon. And what happens is that local depolarization is enough to stimulate the voltage gated channels over here at the next node of RANVA in the axon. And so instead of having to have that impulse travel along the entire length, as we can see here, along the entire length of the axon, instead what we can do is just have local depolarizations occur at each node of RANVA, and the information, the signal, is going to jump. It will appear from node of RANVA to node of RANVA, because right here we'll depolarize and signal depolarization and opening of those voltage gated channels here, signals depolarization and opening of those voltage gated channels here, on and on and on, and it goes extremely quickly. So that is an axon or an action potential. We took a look at continuous conduction, and that's the one that you will especially need to describe. Though what really happens in your body is much quicker using saltatory conduction, though it uses many of the same principles. So again, this is probably the most important video of the entire nervous system. So make sure that you are taking that. I believe there's a quiz on this one. There's definitely a quiz on this one. So make sure you're taking that quiz and asking any questions you have about this.